So I think we can just jump into the next session here. So I'm really excited to present this session. We're going to have 3 MR physicists, and it's really nice to get perspective from some MRI experts. So after a lot of talk from statisticians, these talks will be really interesting. And so we'll have 3 speakers today that are highlighting some of the MR expertise that we have at Emory. And our first speaker will be Deqiang Qiu, who will be speaking about inverse problems, magnetic resonance, quantitative susceptibility mapping. Deqiang, are you able to share your screen?
All right. Thank you, Ben, for the invitation and for organizing this session. I actually was able to turn in a few sections, very interesting presentations. So today I'm going to talk about magnetic susceptibility mapping and its clinical applications. And here's the outline of the presentation. First of all, we're going to go into talk a little bit about the basics of the QSM or quantitative susceptibility mapping. And I'm going to walk through a few clinical applications of these techniques. And lastly, I'm going to talk about the general pre-processing processing pipelines of QSM and the inverse problems involved in this. At the very last, I'm going to share some of our recent work of the where we use Bayesian approach to do sparse image reconstruction for T2 star and QSM. So if you look at how MR images acquired and saved, you can see we usually use MR scanner and acquire some data, and we perform reconstruction and to get the complex valued images. And then we usually take the magnitude images, send those to PAC system for radiologists to review. And then most of the time we threw away the phase images. And here you guys can see a comparison of the magnitude and the phase images acquired at 7 Tesla. And you can see after some processing, the phase images actually show some anatomical details that's not easily visible in the magnitude images, such as the boundary between the globus pallidus and the putamen, as well as these mammalian tracts that's clearly visible on phase images, but not so clearly visible on magnitude images. So all these contrasts on phase images stands for tissue magnetic susceptibility property. So what is magnetic susceptibility? So if you consider a uniform magnetic field, and then you put an object into this uniform magnetic field, this magnetic field is, depending on its magnetic susceptibility property, if it is diamagnetic, it's going to create a magnetization moment that's going to oppose the main magnetic field. Therefore, the net magnetic field within this object is going to be reduced or become smaller. And if the object is paramagnetic or ferromagnetic, then it's going to create a magnetization that's in parallel to the external magnetic field. Therefore, the overall magnetic field it's increased. So if you look at the spectrum of magnetic field, actually it ranges from -1 for superconductors all the way to the 10 to the 5th power for pure iron. And within this spectrum, we can see within this small range, that's where a lot of the susceptibility value of a lot of the biological tissues lies on. And you can see, usually we consider, by definition, actually 0, while the vacuum has 0 magnetic susceptibility, and air also has almost 0 magnetic susceptibility. And if you look at here, the water, because it's actually diamagnetic, therefore it has negative magnetic susceptibility on the order of -9 ppm. And you can see deoxygenated hemoglobin has -5 ppm, and then actually, if you have a lot of iron in the liver, it's going to have close to 0 ppm. So, but when we talk about quantitative susceptibility mapping, we always use water as reference, like point. So anything has higher magnetic susceptibility, we call it positive, to have positive magnetic susceptibility in our QSM applications. So then, so actually these, we see it's sensitive to a lot of things, to iron and to also to myelin, and also to like deoxygenated hemoglobin concentration. So how do we measure it? A typical MR method that's used to measure QSM is really just 3D GRE sequence. So QSM really is a post-processing technique. So if you look at the 3D GRE sequence, any perturbation to the magnetic field is going to cause a phase that's proportional to the perturbation of magnetic field. So it's proportional and also depends on echo time. So actually using this sequence after some processing, you are able to get magnitude image. After some further processing, you can get phase image, and further reconstruction, you can get magnetic susceptibility distribution, which could be used to study different diseases. From this, usually you also use multi-echo GRE sequence. Therefore, you can get T2 star map from this sequence. So back to what is causing the contrast in magnetic susceptibility image. It's really, in the brain, it's really primarily three sources. One is the ferritin. So in the basal ganglia regions, we can, such as the globus pallidus here, you can see it has a very high signal intensity in this QSM image because it has high ferritin level in this region. Also, I mentioned in vein because of it has very high deoxygenated hemoglobin. Similar to the mechanism of both, it has high magnetic susceptibility in the veins. In white matter, because of the myelin, it actually has it's more diamagnetic compared to pure water. Therefore, you see low signal intensity in white matter in the QSM image. So just want to show you a video of this high resolution 0.5 isotropic resolution image throughout the brain acquired from a 7 Tesla scanner. You can see very well a depiction of veins and great white matter boundaries, and as well as high signal intensity in the deep brain basal ganglia region. So of course, because of its sensitivity to iron, white matter, as well as the oxygenation level of the vein, it finds application in many neurological, not many diseases that affects the brain, such as thalassemia, Alzheimer's disease, Parkinson's disease, and myelin that's being studied in multiple sclerosis. I noticed actually there's a parallel session talking about QSM in MS. So also alterations in oxygenation in stroke patients. So this is a study we've done like a long time ago. So you can see in patients with beta thalassemia, thalassemia is a kind of a genetic disease that causes modification of their hemoglobin, and these patients usually requires blood transfusion, which actually increase their iron load in many organs, such as heart, liver, and brain. And actually, if you compare the brain in the thalassemia patients to a normal control, you can see very high QSM values in the corax plexus compared to in patient compared to normal control. And if you actually do a quantitative comparison, you can see the corax plexus has much higher susceptibility value in thalassemia patients compared to control. And actually, these patients also receive iron chelation therapy. So actually, we saw a reduction in globus pallidus. So it's really a dynamic balance between iron overloading due to blood transfusion and this iron chelation therapy. So you can see actually a reduction of susceptibility value in the global pallidus in these patients. And similarly, in sickle cell disease patients, these patients also have a genetic defect, and that affects their hemoglobin. So again, they require blood transfusion and iron chelation, and you can similarly see higher iron deposition in the corax plexus compared to the normal control. So again, you can use the same technique, but if you focus on the vein, the susceptibility values in veins, you can use this to derive index of oxygenation level in the veins. So we previously have done a study in patients with a cerebral vascular disease. If you do some pre-processing to pick up the veins as depicted in figure B here, and then you can calculate a pseudo oxygen extraction fraction. You actually can see in this disease hemisphere, the oxygen extraction fraction is very high. It means it's there's not enough oxygen being delivered to that region, and then that brain region is trying very hard to extract as much blood, as much percentage blood, oxygen as possible. So, so yeah, we were, and after actually a Diamox challenge, we see this OEF actually get reduced a little bit. So, and obviously, actually QSM is very well, because it's sensitive to iron. You can use it to detect microbleeds, and microbleeds contains a lot of iron. And if it bleeds, it get transformed to ferritin or deoxygenated form, so that it can be picked up by QSM. Also, again, QSM can be used for studying the age of a lesion in multiple sclerosis. You can see this a function of the magnetic susceptibility value of MS lesion as function of the lesion age. You can see initially there's increase in susceptibility value, and then which actually return to normal after more than 6 years of age of the lesion. So interestingly, actually, the white matter experience has an isotropic property or anisotropic isotropy in their magnetic susceptibility. So you can actually use something called susceptibility tensor imaging to do white matter tracking. So this could potentially be useful in high field. So, but the authors was able to show actually they kind of overlap with the DTI-based white matter tractography very well. So, I have been showing you a lot of like nice images, but this is the phase we talk about. Actually, you can use the phase image from a GRE sequence to measure the magnetic field perturbation due to the magnetic susceptibility. But this the kind of the phase images you get. Actually, you see a lot of phase wrapping, as well as contamination of the phase due to magnetic susceptibility source outside of your interest, region of interest, in this case, brain. So therefore, you need a lot of processing in order to get something useful. And here is the general pipeline that people use for QSM processing. Obviously, you have to reconstruct the images from the raw data, which is in the K-space, and to obtain the phase image, and then you perform phase unwrapping, and then background field removal to remove the effect from the from magnetic susceptibility source outside of the region of interest, and then finally you perform a dipole deconvolution. So yeah, so there are again many methods used to remove this background field, and this is again one kind of inverse problem. People have developed like methods like sharp V-sharp projection to dipole, and recently total field inversion. So I'm gonna skip some details, but basically after this pre-processing, you can obtain these phase images that's free from the interference from magnetic susceptibility source outside of the brain. So yeah, and then the last step is you can, after you perform the background field removal, you want to perform a dipole deconvolution in order to obtain the magnetic susceptibility distribution chi here. Actually, these you can measure the relative magnetic field perturbation using the phase again, And what you want is the magnetic susceptibility distribution. And in the Fourier domain, they have this simple pointwise product relationship, and this the kernel of this product relation. But you, in a naive like reconstruction, you would just simply divide this relative field image by this kernel, but you can see this this kernel has 0 points, and therefore the reconstruction of this chi is quite ill-posed. And then again, a very like popular trick is you kind of reformulate it as some optimization problem, and then impose sparsity on the solution, and therefore you can generate some pretty nice QSM images from that pre-processed phase image. And actually there's a recent challenge that actually compared different dipole inversion algorithms. I refer the audience to that paper listed here. So, and lastly, I want to spend last few minutes talk about our recent work where, you know, you can do all these reconstruction, but sometimes you want the data has to come from that raw 3D GRE data. So you want to push for higher resolution in order for you to detect small lesions. And one way to do it obviously is to undersample in the K-space in the 3D GRE acquisition. And one nice interesting features compared to other compressed sensing approach is that this 3D GRE signal follows mono exponential function. So we design an algorithm in which we use a generalized approximate message passing algorithm to solve this kind of Bayesian approach. We basically impose a Laplace prior on the wavelength coefficient of the images, and we actually estimate the lambda automatically. Unlike a popular L1 norm-based reconstruction, we estimate the lambda automatically. And so that's one novelty of this algorithm. And another novelty is we, because of the exponential function, it's not linear. So my postdoc Shuai Huang has designed a way to cope with this nonlinearity through generalized approximate message passing approach. So I'm not going to go into the details. So this is our experimental setup. So we scan data in 6 subjects with 6 echoes, and then this is the echo time for the first echo, and then the echo spacing. We acquire very high resolution data at 0.7 millimeter isotropic resolution. And the total scan time for this fully sampled data is about 35 minutes per subject. And then we retrospectively undersample at 10, 15, and 20% sampling rate, and then compare our approach with the L1 norm based reconstruction method, as well as the least square approach. And we call our method approximate messaging passing with the parameter estimate, or AMPPE. And you can see this, the comparison of normalized root mean square error for the T2 star reconstruction. Our methods consistently outperform this L1 minimization least square based method for 10% and 15%, 20% undersampling rate. And this is the metric for the QSM images. Again, consistently outperform on L1 minimization and least square approach. And then this is just the showing images of for T2 reconstruction. So you can see this, the relative error map for 10% sampling rate, 15 and 20. So the last columns are our MP methods. It shows consistently better than other existing methods. Again, this is the like relative error map for the QSM images. So our methods has a better performance compared to least square and L1 norm minimization reconstruction. So yeah, so just conclusion is the QSM is very sensitive to brain changes in many neurological diseases, and there are several inverse problems in the reconstruction of the QSM images all the way at the beginning for reconstructing under sample data to the pre-processing background removal and the reconstruction of the QSM images from the background removed image. So I invite all of these colleagues from the statistics world to work on these problems. And then lastly, we have designed a Bayesian approach with automatic hyperparameter tuning. And we show actually these methods perform better for both T2 star and QSM reconstruction. So yeah, and that's it for my talk. And I want to thank these people from my lab and as well as my collaborators, and then thank the funding source for some of the work. Yeah, that's it.
So our next speaker will be Dave Reiter. Dave, are you able to share your screen? So Dave Reiter, he'll be speaking on advances in quantitative MRI signal modeling and musculoskeletal research.
Can everybody see my screen okay?
Yep.
So first of all, I'd like to thank Ben for inviting me to speak today, and thank you all for attending this session. Before I start, I don't have any disclosures regarding the topic that I'm going to be talking about today. My goal for today is to just provide sort of a broad overview of advanced quantitative MRI and musculoskeletal research, with the hopes that it'll sort of stimulate interest from this particular group in filling some of the unmet needs that we have in musculoskeletal imaging, and in sort of addressing the needs of curing musculoskeletal diseases. So I'll start out with just sort of outlining sort of some of the top diseases that affect the musculoskeletal system. The osteoarthritis, of course, probably being the most well known in the public. Of course, low back pain is something that's very prevalent and costly. And then you've got tendinopathies and diseases that affect the actual skeletal muscle. Many of these are really costly diseases and prevalent that involve sort of a degenerative process. For instance, osteoarthritis affects hundreds of millions of people worldwide. Just in the United States alone, it's tens of billions a year that's spent on management of this disease. And really, this high prevalence and high cost is because there's no real early treatment options that are available for many of these diseases. And that's largely because we're lacking detection of these diseases at early stages. And we also can't really develop therapeutics to address these diseases without having sensitive biomarkers for monitoring therapeutic intervention. And with clinical trials costing quite a bit of money to run on sort of target drugs, having these tools available are quite important for long-term success of treating these diseases. So really, quantitative MRI methods are needed as they provide spatially resolved information that can be sensitized to tissue level properties of the musculoskeletal system, and I'll sort of get into some of the details of that. But as we think about all of these different diseases that I sort of highlighted in the previous slide, there's a variety of tissue traits that sort of dictate the quantitative method and dictate sort of the analytical approach or the signal model that might be used. For instance, skeletal muscle has high cellularity. It's got low matrix in healthy skeletal muscle. It's relatively soft, and its macroscopic geometry is rather simple, meaning that we don't really need high resolution imaging to sort of probe the actual muscle cells. Whereas in contrast to that, articular cartilage has very low cell density. It's not as metabolically active. It's got really high extracellular matrix that sort of leads to sort of more dense like tissue that's more difficult to image because it's the water signal kind of fades away more quickly in conventional sequences. It's also got really complex macroscopic geometry, as you can see from this cartoon over here. You know, it coats the ends of the long bones that have high curvature, and the tissue itself is quite thin. So you need very high resolution images to be able to study or to diagnose disease in that particular tissue system. And each of the musculoskeletal tissues sort of have varying properties on these axes, and of course, you know, those will sort of guide one in tailoring the particular imaging approach. But as we think about quantitative imaging, you know, we can sort of simplify the way we conceptualize this as MRI produces varying contrast where we might select a particular imaging parameter that we vary over a range, and we have a particular signal model that we would fit to these different images to generate biophysical parameter maps. Some examples of these that are used in musculoskeletal imaging are listed here in this table. And just to highlight that, you know, a lot of these are sort of very simple exponential expressions and lend themselves to, you know, sort of easy modeling. But the models themselves, of course, arise from the physics behind the sequence that's selected, as well as the underlying physical picture that we expect from the tissue system that we're studying. And so I just want to use cartilage as sort of a tissue to illustrate some of these concepts as we consider, you know, how we can improve quantitative imaging. As I mentioned, cartilage, you know, deteriorates with osteoarthritis. You know, it's really characterized clinically as thinning of the articular surface. So this is sort of a zoomed in cartoon of what the cartilage ultrastructure looks like on the surface of the long bone here. And you can see that it depicts these sort of fibrous collagen fibers that are oriented perpendicular to the subchondral bone here. And as we move away and towards the surface of the cartilage, these collagen fibrils are oriented more parallel. So there's a very complex structure even within the through the thickness of the tissue itself, making it more challenging to image and to sort of characterize. At the microstructural scale, compositionally, it's comprised of primarily type 2 collagen, which is a triple helical molecule that's quite rigid. It provides tensile strength for the tissue itself. And it's also has attached to it these sort of more wavy, mobile proteoglycan molecules, which are hydrophilic, and so they draw fluid into the matrix and provide its viscoelastic properties. The important piece of this is that at early stages, before we start to actually measure changes in thickness with osteoarthritis, we start to see a depletion of the concentration of these macromolecules. So they serve as sort of biomarkers of early stage osteoarthritis. So being able to detect these changes in concentration in this very difficult to image tissue would provide valuable information for early diagnosis and for monitoring of therapeutics. So you can imagine that water, which is really our molecular probe of MRI, exists in these different environments that that microenvironments near these different matrix constituents. And the vast majority of cartilage is water by wet weight. If we think about the mobility of water sort of in the bulk phase, where it's sort of moving around away from these molecules, its relaxation would be considered to be quite slow. And as we get closer to the matrix molecules like proteoglycan, which is not quite as rigid as collagen, but still a macromolecule, relaxation of water it relaxes faster. And then within the collagen fibrils itself, it would relax quite rapidly because it's near a rigid structure. So you can imagine that our signal model, which we showed originally as just a simple exponential, might actually be more appropriately modeled as a sum of exponentials in order to capture some of the complexity of this matrix environment and the water that's sort of interacting with it. So we could explore a variety of different signal models that might provide extra sensitivity to degenerative changes. And the way we tend to go about doing this is a mixture of using very high quality signal from explant studies, where we can stick an explant in a research system and we can get really high sampling of the parameter space with really high signal to noise and test complex models. But ultimately, we know we have to simplify our models as we move towards in vivo imaging, where our signal to noise is not as great and our sampling is poor. And this is really where the divide exists and where we really need help, particularly from a community like this to provide input and solutions. So starting out, I'll sort of use this graphic to depict what type of quality data I'm talking about when I describe some of these models. With this multi-exponential model, which it's an ill-posed model, but it can be inverted using some numerical approaches like non-negative least squares with regularization, which is what we did here in these tissue explant studies. You can see the structure of the T2 histogram is quite rich in a healthy sample. And then when we selectively deplete proteoglycan from the cartilage, you can see that some of these structures that we attribute to water interacting with more rigid matrix structures, some of these start to diminish. And in fact, if we quantify these, what we consider to be sort of water or proteoglycan associated water pools with biochemically derived proteoglycan content, we can see we get a really good correlation, suggesting that there's value in some of these more sophisticated models. As we start to diminish our signal quality and our sampling of these echo decays using microimaging here, which is shown in these two maps of young and mature cartilage, and using a more simple model, a bi-exponential model, we can still capture signal from this proteoglycan-bound pool. And we can see that when we compare it to a gold standard quantitative method like FTIRIS, we're able to still see a good correlation between our MRI-derived water pool that we think is associated with proteoglycan and the actual proteoglycan content. Now, when we start to think about extending this with a multi-echo spin echo imaging experiment in vivo on a clinical system, we can see that from these simulations that we performed, numerical simulations looking at sort of the admissibility of fitting a bi-exponential model, our signal to noise requirements in order to be able to resolve these with very good accuracy or precision is pretty high and prohibitive for being able to do this in a very thin layered structure like cartilage. And so, we can think about, to approach this, we can think about more simplified parametric models like the stretched exponential, which can be thought of as a distribution of relaxation times. This is the inverse Laplace transform of the stretched exponential. And you can see that the multi-exponential fit to these cartilage samples sort of resemble, or I should say the stretch exponential kind of traces the envelope of this multi-exponential fit using really high quality data, high signal to noise, suggesting that this model sort of captures some of the same information that we might be after for detecting proteoglycan changes. And the stretched exponential parameter also correlates quite nicely with biochemically derived proteoglycan. And if you actually compare it with just the bi-exponential model, when you look at the fit residuals between the data and the model itself, we see that we have a much better representation of the data with the stretch exponential model.
Hello.
Yes? Okay, I'll keep going. And if we think about extending this type of model to clinical imaging conditions, where we've got lower signal to noise and we've got longer echo times, you can see that the accuracy and precision of this more parsimonious model of multi-exponential behavior is more robust. Just to sort of illustrate some other approaches, though, that step outside on the acquisition side of sort of the more classic ways of measuring relaxation, there are other approaches that use gradient echo sequences that can be much faster, higher resolution, and produce higher signal to noise. And this is an example where spoiled gradient echo and balanced steady state free per session imaging can be used to quantify bi-exponential T1 and T2 relaxation. This was originally developed for myelin mapping in the brain, but has been extended to studying cartilage, and has actually shown in some studies to be sensitive to changes in cartilage with osteoarthritis, as shown in this study by Liu et al. What we see here, though, is that the imaging time is still quite long. It's 17 minutes, and the resolution of the image, the pixel resolution, is still about double what you would desire for doing real imaging of cartilage. So that's a big part of that is, also you can see that although there's a statistical significance between these two groups, that the difference isn't really that stellar and might not really have that much diagnostic value. So to sort of improve upon this, one of the things that can be considered is that this model, the signal model, is actually quite sloppy and difficult to invert. And so that's been approached recently using sort of a Bayesian type of approach for parameter estimation of the bicomponent model from this acquisition. And just to sort of illustrate some of the highlights of that, the imaging resolution can be pushed down to something that's actually feasible for looking at layering the different layers of articular cartilage in the knee joint, for instance. The imaging time is still a bit long, but the Bayesian approach definitely shows a significant improvement in parameter estimation compared with sort of the standard global optimization approaches that have been used traditionally in the McDespot approach. So I hope through this talk, I've been able to sort of motivate that there's a lot of work that can be done in terms of model selection for improving the sensitivity of quantitative imaging in the musculoskeletal space. There's a lot of work that can be done in extending novel sequences to accelerate acquisitions and to improve the resolution and have more feasible physical models. And certainly there's one of the big needs is to have these more practically usable. We need better estimations and we need faster scanning. And as Deqiang talked about in his talk and his applications in QSM, certainly we can do better with improvements of estimation of the actual parameters. We can also accelerate. We also need to do better at applying acceleration methods to the data space or to invert the data space into image space. We can see from this review here that in the space of musculoskeletal imaging, some of these advances that are used in the brain are really underutilized in applications of musculoskeletal imaging. As you can see, that the compressed sensing, as an example, is barely ever applied to cartilage mapping. And certainly estimations from the data space, from undersampled data space all the way to the parameter space, there is really very little that's been done in that space for musculoskeletal imaging. So I'll stop there and I look forward to questions at the end of the session. Thanks.
Our next speaker will be Candace Fleischer, who will be speaking about metabolic and thermometric brain imaging with magnetic resonance spectroscopy.
Great, thanks, Ben. And thank you for inviting me and also for organizing this session. So I want to talk a little bit today about magnetic resonance spectroscopy, and I want to introduce this technique as well as discussing some of the research and clinical applications. So to give you an overview of the work that my lab does, we focus on magnetic resonance spectroscopy for quite a few applications. We do a little bit of work in the data reconstruction space. So I'll talk a little bit about this, but MRS as a technique is relatively slow and low resolution. So we work on improving the acquisition and post-processing as well. We're also looking at metabolite changes in a number of different cohorts, but largely related to inflammation and chronic pain. So it's been demonstrated that both of these can induce changes in local brain metabolite concentrations. And so we're trying to explore this a little bit further. Another big focus area of my lab is to look at brain thermometry and thermal modeling. And I'll talk a little bit about this today. But brain thermometry can also be used from this MR spectroscopy technique. And lastly, a big focus is developing new imaging biomarkers using this method for a wide range of diseases. And what I'll focus on today is just to give you a snapshot of how we can do this with the case study of brain injury. So it's well known that brain injury causes structural and connectivity changes. Of course, we know about those, but we also see that the brain can undergo chemical changes largely due to metabolism and temperature changes largely due to alterations in hemodynamics. So I want to talk a little bit about how we can use this technique to explore both of these. So how does it work? So MR spectroscopy uses the same principle and is effectively in vivo NMR spectroscopy. And of course, we know this is the same principle as what we see in MRI. We just use the technique slightly differently. So for general MRI, we know we need a magnetic nucleus and a magnetic field. And by putting that nucleus, such as a proton or a hydrogen nucleus, in that field, we get this energy splitting. And what this results in is this energy difference, which is what we exploit when we do any sort of magnetic resonance experiment. So generally, when we're looking at most magnetic resonance techniques, including the ones that we just heard about, we're focused on the water protons. And when we're just focused on water protons, we assume that the energy of those protons or those nuclei are identical. And that's a good assumption. But if we want to look at protons in other molecules, things like amino acids or proteins, we actually know that there's an energy difference. So for example, a proton in an amino acid is going to have a slightly different energy or frequency than a proton in water. And we can use this to get a magnetic resonance spectrum. So why are the energies different? And this is due to a principle known as magnetic field shielding. So in MR physics, we often think of the nucleus as being alone, but of course we know that it's surrounded by electrons, and those electrons effectively shield the nucleus from that external field. And so what effectively this does is changes the magnetic field experienced by the nucleus, giving rise to different frequencies. And it's those frequencies that we can look at with a magnetic resonance spectrum. So instead of an image, we're actually getting a spectrum where each peak in the spectrum corresponds to a different nucleus or a different metabolite. And our x-axis in these spectra are always frequency. We report it in a chemical shift to normalize against the strength of the field. But each of these peaks and the area under the curve of each of the peaks can give us an estimation for the concentration of the metabolite. In my lab, we primarily focus on the brain, but you can do this throughout the body as well. So MRS is an FDA-approved method. And again, like I said in the beginning, because of how we acquire the data, it's relatively slow. And because we're looking at all of the other metabolites other than water, we're looking at things that are about 1,000 times less concentrated. So we get much lower signal than we would, for example, in a structural image. And it is used in the clinic. There are quite a few different implementations, but the most common is to look at a single region at a time. We call this single voxel spectroscopy. And again, in each of these regions, we see we can actually get a localized spectrum with localized metabolites. We can also do multiple voxels at the same time. We call this MRS imaging or MRSI. And again, each of these green boxes corresponds to a volume element, which will each produce an individual spectrum. And then more recently, we're able to do this across the whole brain. So we can actually get whole brain metabolite maps. This is largely limited to research, again, due to the length of the scans and some of the challenges, but we are able to use this in a lot of research applications. So what does this look like in the human brain? Largely, the healthy human brain will have peaks that look something like this. So the largest peak in our spectrum is N-acetyl aspartate. This is a neuronal marker. We also have choline and creatine. Myoinositol is a marker for neuroinflammation, a surrogate marker. And then we can also look at neurotransmitters, things like glutamate and GABA. So the work in my lab is largely focused on varying degrees of injury, and I'll focus on traumatic brain injury as a case study here. And we look at this across the full spectrum. So everything from subconcussive or concussive. So this is largely what we see in athletes. We've been doing some studies in collegiate athletes. We also have used animal models to look at more severe traumatic brain injury. And then all the way down to a very severe case where the person has a loss of consciousness. We're doing some studies in coma patients as well. So let me give you a couple of examples on what this looks like. So in our piglet model of traumatic brain injury, we looked at this over a period of time, but 24 hours post a controlled traumatic brain injury. If we look at the seemingly healthy or the contralateral side of the brain, the spectrum actually looks fairly similar to what we saw in healthy humans, where we have a large NAA peak and then both a choline and a creatine peak. But if we look at the region of the brain that's injured, we often call this the ipsilateral side, we see that we get a significant loss in both of those peaks, all of those brain metabolite peaks indicating a loss of neuronal function. We also get the presence of lactate, which depending on how we acquire the data is sometimes inverted. And lactate, of course, is a sign of hypoxia. So this is one example where we can use MRS to look at localized changes in the brain as a marker of injury and also brain health. If we look at a more severe case, we're currently actively recruiting studies we're recruiting patients in a study with the Shepherd Center. We're looking at patients who are in a coma, either a minimally conscious state or a vegetative state after traumatic brain injury. And I will say all of these patients so far actually have been due to automobile accidents. I can look at a couple of examples. So we have a 20-year-old male who had a diffuse traumatic brain injury with loss of consciousness. See this very dramatic decrease in this N-acetyl aspartate peak, increase in choline. So we get a loss of neurons across the brain. We're primarily looking at this in the brainstem, but of course we can do this in other regions as well. This patient actually hasn't emerged from the coma. The decrease in NAA here is often indicative of fairly severe injury. Contrary to this, we have another case where we had an 18-year-old male with very severe TBI, a portion of the brain was removed. We actually retain this NAA signal given that there was a good amount of neuronal function, and this subject actually emerged from the coma. And so we've recruited about 25 patients so far, but we're really trying to understand how brain metabolism and local chemical changes in the brain can be used as prognostic markers in these more severe patients. Because of course, that is the big question in coma is the patient going to emerge? So the other thing we can look at are temperature changes in the brain. And this is much less common. We actually don't have a method to do this currently in the clinic. Other than invasive implanted temperature probes, which you imagine is not very common even in very severe patients. And brain temperature, we believe, is regulated by very high cerebral metabolism, and cooling occurs due to heat dissipation from cerebral blood flow. So the blood that comes to the brain from the heart is largely cooler because the body is generally about 1 degree cooler than the brain. So there's a good amount of evidence to suggest that brain temperature actually is very important for recovery after a wide range of injuries. This is one example from stroke where body temperature on admission was associated with worse outcomes. So higher body temperature led to worse outcomes of the patients. And this is further supported by evidence that cooling, also known as therapeutic hypothermia, actually leads to some form of neuroprotection. So the mechanisms are not entirely clear, but the idea is that if we can cool the body and subsequently the brain, we can actually protect the neuronal tissue. So how can we measure this using MR spectroscopy? Normally, when we look at MR spectroscopy, we're mostly concerned with the very generally the area under the curve or the integral of the peak, which is a surrogate for concentration. But we can also actually look at the peak position itself and the frequency. And it turns out that the frequency or the chemical shift of water is largely temperature dependent. So as temperature increases in water, the hydrogen bonds are broken, which leads to an increase in electron shielding. And what happens is that water peak actually shifts upfield to a lower frequency. And so we can use this chemical shift difference as a metric for temperature. And luckily for us, it happens to be very linear direct relationship if we look at that chemical shift difference as a function of an external temperature probe. So we're able to implement this in a number of different scenarios. We're able to do this on a whole brain temperature scale. But as you can see, even though we see some consistent patterns for gray and white matter, the data is fairly noisy and we do get some corruption, especially around the surface of the brain. So to complement these experimental thermometry metrics, we're also doing some brain temperature modeling. So by using the first principles of energy and momentum and mass conservation, as well as heat transfer equations, we're actually able to model on an individual voxel level the temperature of the brain. And I'll spare you a lot of the details of the model, but most simply, we look at this across 3 domains. We look at tissue, arteries, and veins. And then we also look at the heat transfer modes of conduction, which is heat transfer between surfaces, advection, which is heat transfer due to fluid flow, and convection, which is a combination of the two. And by fully conserving our energy, mass, and momentum, we're able to predict temperature using just first principles without any empiricism. So the way that we did this in humans is we actually acquired tissue and angiography and venography or vessel structure. And using those individual tissue and vessel structure maps, we're actually able to predict and model metabolic heat, which generates heat, and cerebral blood flow, which dissipates heat. And using the combination of these two, we can actually predict temperature on an individualized basis. And so what we observed was that the absolute temperature, as well as really specific local gradient patterns of temperature across each of these subjects, was distinct. And we also observed that these modeled temperatures, using no empirical data other than the tissue and vessel structure maps, aligned very well with our experimental MR thermometry, really supporting this combined experimental and simulated approach. And because of these differences, what we propose is that personalized medicine, which of course we know is emerging in contemporary medicine, particularly with thermometry, may be really important. So generating these personalized temperature maps for each individual may be really a crucial step when we're translating this to the clinic. So we're also extending this to patient studies. We've done this in several patients who have had a stroke. Again, we can simulate that by blocking a vessel, which is what happens in an ischemic stroke, and we can model the temperature as a result. And so we can look at temperature changes across the brain, and we can do this in a very dynamic way for a number of different cases where we model changes in blood flow and then produce the subsequent temperature maps as well. So where we think this is headed is, for a lot of these injuries, we really are very aware that structural and functional changes occur in the brain, especially after stroke or traumatic brain injury. But what we believe is that this extra component of metabolism, which is largely influenced by things like inflammation, is actually this additional piece that can provide a lot more prognostic information for a lot of these cases where we don't have good prognostic biomarkers. And metabolism can really add to this picture of understanding what's going on for individual human brains. We're also implementing this combined experimental and simulated approach to thermometry in a number of different patient cohorts, including stroke and variations of stroke, as well as in patients who are undergoing cooling for cardiac arrest. And again, we really believe that it's this personalized temperature approach that will allow us to make strides in the clinical setting. So with that, I'd like to thank you all for your attention. Of course, my lab and collaborators, as well as our funding. Thanks.